Good afternoon, everybody. I'd like to start by saying thank you to Invivoscribe for inviting me to talk on behalf of our lab and the work that we've done over the last, say, 4 months now, mostly carried out and directed by two talented people here in the audience, Dr. Meenakshi Malhotra, our Assistant Director, and Asha Reddy, one of our supervisors. This work has been involved in analysing and transitioning over to NGS-based analysis for TCR clonality, and in particular the focus has been on looking at T-cell disease in MDS because at Mount Sinai we do a lot of T-cell clonality testing in that disease. I'd like to start by saying these are my conflicts of interest. There's some pathology, departmental negotiated salary support for myself from Semaphore, as well as I own some stock in CGIX. So I usually start with this slide when I'm talking to residents, and while most of us in this room here are very familiar with the pros and cons and problems that we have interpreting gene rearrangement analyses in patient specimens, we're also all very familiar with what's on the left side here, the technical aspects, and how over the years transition from gel electrophoresis over to fragment analysis and now on to sequencing NGS-based. During this transition, we've increased not only the specificity but also the sensitivity, so that we're hoping at some point to be able to go down to able to use it to track for the presence of a clonal rearrangement at very high sensitivity. When I took over as directorship at Mount Sinai of the molecular lab, they were performing these assays for IGH, Ig kappa, TCR gamma, TCR beta using gels. And I made the decision I was going to bypass fragment analysis and just go straight to the NGS platform. And that's what I'm going to share and talk to you about today. So this slide is not so much for me to transition and talk to you about all the different rearrangements that you can get, but more to focus on the fact that we are going to be talking about gamma and beta. And in these two genes, just to remind you that in the normal ontogeny of T cells, as part of the process of their maturation, the TCR gamma gene undergoes rearrangement first, and then if a functional rearrangement is not actually made and produced, then the cell will go on to rearrange the TCR beta gene. The other thing to notice is on account of that also, you get much less diversity in the TCR gamma rearrangements than you see in the beta gene because of the resulting VDJ rearrangements. But you can also get in the TCR beta gene, you can get some incomplete rearrangements between the D and the J segments only. And you'll see that in some of the data that I'm going to talk about. In this transition phase where we are, we're still in our optimization phase, getting close to move into our full validation. In this optimization phase, we've run a number of cell lines, dilutions of cell lines, specimens. We've compared results between our specimens that we've already run by our gel-based methods, both peripheral blood as well as formalin-fixed paraffin-embedded material. And we've assessed different inputs of DNA. I'm not going to go over some of that data, but suffice to say we've settled on 100 nanograms starting input DNA. What has been, as for most of these NGS-based assays, the dry bench headache is how are we going to establish our calling criteria. And we've actually, I think, pushed ourselves to get very close to what we need. This is based on a series of publications that were both for TCR gamma. Also, we use some of the immunoglobulin rearrangement-based publications to also help guide and inform how we might look at these polyclonal backgrounds. We've also been given valuable suggestions from Invivoscribe, so please reach out to your specialists from Invivoscribe if you're thinking of bringing this in, because they really are helpful and supportive. I've listed a few of the publications there that we've done, but that we've used, and there were others. So this shows you for us what I think we're getting close to now, our final criteria. We insist that each sample has at least 50,000 reads, that we have our first cuts in that it emerged read clone, meaning that, and the software is provided by Invivoscribe to define these merged read clones. They are considered as a merged read group because they only differ by less than 3 nucleotides in their sequence or length. If we don't have one of those that's more than 2%, it's called polyclonal. If we don't have a difference between, as you go down the frequency in terms of these merged clones by at least 2x from one to the next to the next to the next, it's called polyclonal. If we don't, then we evaluate our polyclonal background based on the last merged read clone that did not show a 2x change to the next consecutive one. Next category is only the samples now that pass these 2 criteria get considered further, where you have to have at least one merged clone greater than 4.5%. As you start looking at some of these sequencing data, you'll realize that that 4.5% is actually not a very accurate measure, because you can't control in clinical samples the denominator in calculating that frequency. And we have as yet to try, and we'll start trying now by adding in a standard DNA that will allow us to then give a more accurate percentage. The next thing we also need to have to move on is to have a clone that's at least 4 times the frequency that is found for the polyclonal background. For TCR beta, we've actually lowered that to 3x. If neither is satisfied, samples called polyclonal. If either one of them is satisfied, we've decided to call it polyclonal with restricted VJ usage. Obviously, if we pass all of these, we're going down now into the realms of are we calling them monoclonal or oligoclonal, and that's based on the number of clones that we see. Just to give you an example, we've run quite a few cell lines, both internal and as available commercially. We ran them all straight. This was not just done once, they've been done several times. These are the clones that we found, and based on our criteria, what would we call them? You can see for gamma, it's been pretty straightforward, but beta, we've had some that we've called oligoclonal, mostly because some of the clones are actually, the next lower down ones are actually quite lower, but they still satisfy a 2x above the next consecutive clone. We may have to address that in our calling strategy, and that's the same case as when we start doing dilutions now. You'll see that we feel, based on dilutions of these cell lines, that we're establishing, at least for gamma, our limit of detection with this calling criteria at 5%. But beta, again, we're having issues with, especially when we come to use this cell line where we have polyclonality versus monoclonality. It could depend on the actual clonal rearrangement that's in this particular cell line. So moving forward, we're just going to call samples of clonality, not tracking yet. Now, if we look at the samples that we've done 58 so far. Some of them were peripheral blood, some paraffin embedded, and this is where we started to get a bit of a shock. So for the ones, if we look at what we call our gel-based, not clonal, 26 samples, all of them were called polyclonal. We're good. Some of them we called indeterminate, were kind of a mix between oligoclonal and polyclonal, but when we got to the ones on our gel-based assay that we called clonal, where it was highly subjective as to the intensity, and based on the gel you don't have the resolution to separate clones that may differ by a single base pair in length, or 2 or 3 even, we started to find a number of calls now drop down into this polyclonal back category or with restricted VJ usage. They no longer became as clearly clonal populations of cells. Of note, our paraffin-embedded material in both cases performed very well and consistent with what we expected. If we go to beta, a similar story happened, except we had an additional surprise here where one of our so-called negative samples now became monoclonal. We can look into that, and well, we think we know why, but you know, these are the sorts of surprises. Now we have insight into the actual sequences. Again, we had a large number of them. that dropped down in their estimates of clonality. And it's important to note that 7 of the samples that were in here that were actually dropped, they were considered before clonal both for gamma and beta, and now they are both polyclonal. So they have no evidence of clonal T-cell disease based on this assay anymore. I think it was somewhat fortuitous, or I don't think it was by design actually, but it turned out that 2 samples that we had belonged to, and there's 2 patients, I should say, that we had 2 samples for. One of them was a PTCL, where first of all we were actually asked to analyse based on the peripheral blood, and if you use our calling criteria both for gamma and beta, they would be considered polyclonal. A couple of weeks later, there was a diagnostic biopsy performed, and in fact now we can see that in fact the specimen clearly has monoclonal rearrangements for TCR gamma and for beta, and I've written here the families that were involved and the percentages that we obtained for these specimens. This is FFPE specimens, so you can see that they work very well. What we found, if we went back and looked at this specimen now, is that in fact that clone was there, but at a very low percentage. Again, I can't guarantee that that's the percentage because we don't have the denominator properly defined yet. However, in the TCR beta, one of them was not detected anymore. The second sample is a bit more straightforward. Again, we had a diagnostic biopsy. This time monoclonality was defined with the families. This was an ATLL. And 3 months later in the peripheral blood, we actually see the clone still there at a reasonable percentage, but according to our calling criteria, we would in fact be calling them PCR VJ, that's with the restricted VJ usage. So you'll find then that this is actually, the clinicians have started to send some peripheral blood, not for this yet, but in general, and we're itching to be able to tell them, yes, that clone is present still or not. So we're rapidly trying to undergo validation for this down to a lower level. Now I want to switch to what was the topic, but we had to come through that process to be able to talk about MDS and what's being done at Mount Sinai. These are patients who in the past who have been called MDS with T-cell disease. And this was based on the fact that they showed T-cell clonality by our previous gel-based assay. And I think what we're going to have to do now, based on some of the results that I'm going to show you, that some of them will no longer be considered with T-cell disease. I don't need to go through what MDS is, but suffice to say that immune derangement is evident in MDS. And in fact, it's even been suggested to contribute to the pathogenesis of the disease and allow for clonal expansion. In fact, some of the MDS patients do show expansion of cytotoxic CD8 positive T cells, which can be harmful to hematopoiesis. And it's thought that some of, in particular for TLGL, these proliferation of cytotoxic T cells manifests itself as neutropenia, hemolytic anemia, and also some thrombocytopenia. And in fact, it's been reported that you can see presence of these such clonal populations in about 50% of the bone marrows of MDS patients. It's also been suggested that such disease or these proliferations can actually be improved if the side effects of it by immunosuppressive treatment. And in fact, at our institution, some of these patients with T-cell disease are treated with intravenous immunoglobulin, IVIG, and this is led by Dr. Silverman at our institution. We have a clinical fellow who's now involved in this project, and also Shafanaz Hussain, one of our hematopathologists in this. So when this became apparent, Dr. Silverman became very excited about how now we could look into some of these. And we started off with 9 patients. I have to tell you that these are independent specimens from what we had done in our calling criteria. And you can see I've categorized them here in terms of the WHO classification, I've given you some risk characteristics according to IPSS, the cytogenetics, mutational profile. Importantly, the duration of treatment with IVIG for these patients. They got included in this because they had at least one sample that was positive for T-cell gene rearrangement. We had longitudinal sampling of these patients, and we also then went in and performed the NGS-based assay. We performed it on 2 to 4 specimens per patient. And I'm going to start, first of all, with the description of what we saw with this one patient who was already considered to have LGL. So just to orient you here on the way I've done some of these figures is that if you look at this patient and you go from this is from 2014 to 2018, based on the gel, they were all considered positive for clonality. Here's the TCR beta, they were all considered negative. The NGS, based on our calling, showed that these samples are consistently called polyclonal, but on the other hand, we did have identify one it was comparable with calling a positive biclonal, but over time, over these 4 years, they would drop down in their categorization as monoclonal, but we could still see the clone present. And this is an example, this is what it's drawn as a percentage over time from 2014 through to 2018. This patient has actually been on IVIG for 55 months. So whether that's responsible for lowering that clonal level and keeping some of the side effects and impacts of these elevated cytotoxic T cells that are circulating remains to be seen, and we'll investigate that further now. The next group, a lot of data on one slide, but I wanted to point out a few things. These 5 patients all had refractory cytopenia with multilineage dysplasia, classification of MDS. And what the surprise was here was that all 4 of these down here, they had positive, except for this one was indeterminate, positive, 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 positive by gel. Not one of them now showed monoclonality at any point. So, but interestingly enough, as I've shown in this box here, we could still see the same clone family and actual sequence in a variety of them over time, but it didn't reach the level at which we could call it a clonal population. So we have to be cautious in how we're looking at these, calling them and interpreting them. The patient and you'll notice in these patients that the vast majority of them, except for the first one and the third one perhaps, at least for the first one, they did not get treatment with IVIG for very long. The first patient actually was monoclonal for gamma, and you can see the families here and the rearrangements, and they drop down over time. This patient was treated for a long time with IVIG. Lastly, 2 patients with refractory anemia with excess blasts, and I apologize, this one is around the wrong way, is that in both patients had calls to be called monoclonal. They satisfied those calling criteria, and over time, this patient was actually treated for treated for 45 months, and over time, this is from 2013 to 2018, over 5 years, the clones were still clearly evident, but not adequate to be able to call them monoclonal. And the same thing happened now with this patient, patient 9, who was called monoclonal. And you can see now, at least for this TCR gamma rearrangement, over time now his clone has risen, and it turns out that he's actually stopped IVIG treatment now. So we're still now trying to correlate the exact times at which the treatment is stopped and started with what we see at this level. So in summary, what we have for TCR clonality and MDS, our method of transitioning over to NGS has revealed that we can actually see these clones low level in a longitudinal manner in these MDS patients, often up to 5 years that we can still see the same clones present. Out of 9 of the cases that we've studied to date, 5 of them had polyclonal T-cell populations. And these were, though, again, we could still see small traces of it over time. And they tended to be people who had not been treated for an extended period of time with IVIG. In 4 of them, we had at least some monoclonality detected, and again, at lower levels, we could again follow those for multiple years. What we're proceeding with now as part of this study, separate from the clinical validation, is to include additional specimens for these 9 patients. For some of these, we have up to 9 to 10 specimens that we will analyse and correlate the exact time point with the treatment that the patients had. And we're going to expand the cohort to reevaluate clonal T-cell in MDS. So what we originally thought as MDS with T-cell disease may in fact not be. That remains to be seen how we redefine that category. And with that, I'd like to thank you for your attention. This is a picture of our lab group back in the lab. We've obviously had a little addition to the family, and thanks also to Lewis and Jonathan and Shafanaz for their contributions. And thank you.